pequeña presentación o nos vamos derecho a que él comience?
No, sí, presenta.
No podemos dejar de ver tu hermosa voz.
Ay.
We cannot stop listening. Beautiful Lina voice.
Oh, thank you. So, Misha, I'm going to introduce you to the audience. So I will just read the text that you sent me. So I will start with that. So it is my great pleasure to present you the talker of today, that is Mijailo Azarc. He's a senior doctor at the Drescher Group, and I'm going to read you some of his previous studies. So he studied inorganic and coordination chemistry at the University of Kyiv in Ukraine. Then he worked at the University of Göttingen in Germany, and then he made his PhD in biophysical chemistry at the University of Constance in Germany, with the topic distant measurements by EPR. Then make a postdoc in Leiden in Netherlands, working with high frequency EPR. And since 2016, he's a senior scientist and lab manager in the Drescher Research Group of the Department of Chemistry in the University of Constance, Germany. So thank you so much, Misha, for being here. Go ahead.
Okay, thank you very much, Lina, for the introduction and the invitation for you and the rest of the organizing committee, so to say. Well, so this is the talk, as Lina asked me, about state of the art in beer. And so there are many techniques, or quite a number of techniques, to study structure of biomacromolecules, and DEAR is one of those. Now, and in this talk, I'm going to talk about the technique, what can it and how to apply. But first, now, if we recall the techniques that are already known and more widely used, say X-ray crystallography delivers basically the full structure, but this has to be crystallized, and these are not physiologically relevant conditions for the structure. Then we have also NMR spectroscopy. Well, this delivers the full structure as well, and then in solution, but the limitations is then the size. Very large molecules or molecular complexes cannot be studied because they rotate too slow and the lines are too broad. Then we have cryo-EM. This is a novel technique, or recently recognized by the Nobel Prize. It also delivers the full structure. But here the sample preparation is really cumbersome. Then we have also FRET. These delivers one distance constraint per molecule, but then in solution. So what is DEAR? This is the topic of this talk. Now, first of all, if you read literature, then you have to know this one. DEAR is Peldor. And these are the acronyms for two different, for the same thing. DEAR stands for double electron resonance, and this is pulsed electron double resonance. So this is the same thing. This is just one technique within a larger collection of spectroscopy techniques where I'm active in. It is electron paramagnetic resonance. Let me take the pointer. Exactly. So, and with this technique, we can also determine distance constraints in biomacromolecules. But this is essentially a spectroscopy technique. And what we measure or we determine the distance between two paramagnetic centers that are dipolar coupled. And this is the way as we normally represent these. The two arrows are the two electron spins in this case, and these ovals indicate the dipolar coupling. So the technique essentially measures dipolar couplings, not the distances. But then these frequencies of dipolar couplings are translated to distances with this rather simple formula in mind. So the frequency that we measure is inversely proportional to the cube of the distance. Okay, so here is now this typical figure, typical image we are going to work with through the whole presentation. And let's look now at the typical data. What we measure in our experiment, or how the data look like, we measure the oscillation of the spin echo. So here is, for example, of these oscillations, and they are then for a distance of 3 nanometers. Yeah? If the distance becomes larger, say 5 and 7 nanometers, then these oscillations become slower. And this is exactly this formula on this slide. So the smaller the frequency, the larger the distance. Yeah, and here, even for 9, it goes even larger. And here is also the range. What is simulated here, roughly from 2 to 10 nanometers. Within this range, we can determine the distance, and we can also determine a number of distances. In fact, if we have, say, 2 peaks here, we will have 2 oscillations in there. Well, then, in order to realize this in the biological matter, which is essentially diamagnetic, we have to introduce these unpaired electrons inside. So we want to have at least the situation like this. And then these 2 arrows, they represent unpaired electrons. And and this is then a spin label, a small molecular fragment that bears unpaired electron. And there are many of those that are currently available and are currently used. Now let's review them. Here they are. I start here on the left with the nitroxide. This is in fact the first spin label ever used, ever designed, a small stable radical, stable and ambient conditions on air as a powder in solution. So just stable and very handy to use. Now here is another example. This is a metal complex, here shown a manganese inside, but it can be also copper or gadolinium as well. So in general terms, any metal that bears unpaired electrons can be used as a spin label, but because of the properties, some of them are preferred over the others. Then here, the third tile is a trityl radical. So carbon-centered radicals as well, as long as they are stable, and here it is stabilized by the bulky substituents, here can be used as well, and they have also some advantages. The most interesting tile is this one. The molecule that is shown here is not a radical. It is a di-eosine, and this di can be excited by the laser to a triplet state. The triplet state is paramagnetic, and this is then the radical or the paramagnetic center that is induced just at the moment you want to have it. Well, so these are the different types of spin labels, and let's now review also the methods to introduce them into the molecules, basically to realize this. Yeah, and I will review this. Well, the method to introduce is called spin labeling, and more so it is called side-directed spin labeling, because what we do by this, we insert our spin labels deliberately at the site of interest. And I will review this now for proteins, but it is equally applied for amino acids. So the first approach is here. If we are about to have a short peptide that can be synthesized in a solid peptide synthesis, then we just take a specific amino acid that already bears an unpaired electron and then insert it at the position we like. If it is further possible to take this short peptide and to ligate it to a larger protein, then we have the whole large protein spin labeled. So these are some typical representatives for the amino acids that are schematically shown here. Talc is the widely used one as a commercially available one. So this amino acid can also look like this, and this is Prof, which is proxyl-labeled phenylalanine is also the development of our group. So these were the short peptides. What about large proteins? So they are, of course, of interest. Now here, the approach can be taken in general terms exactly the same. We take an unnatural amino acid and incorporate it inside the large protein. And these approaches are known. To realize this, you need the non-canonical amino acid, suitable orthogonal synthetase and orthogonal tRNA. And by this, you create this important unit that then, by the translational machinery, given that mRNA also has an orthogonal codon at just the position of interest, there is then synthesized a nascent polypeptide, which has then an unpaired electron, exactly the desired position. And this is, of course, a very elegant way, because you let the translational machinery work for you. And here is the development of our group, and in fact, the first non-canonical amino acid that bears an unpaired electron, and with this unpaired electron can be introduced in the protein, called acyl-K1. So it is spin-labeled lysine. For those who are familiar with amino acids, this is the lysine, and this part is then the conjugator spin label. So indeed, as you have seen, it is possible to spin label protein during the synthesis or expression, but it is also possible, in many ways also easier, a post-synthetic modification. It is this group of methods, number 3. In this group of methods, a specific residue is targeted, and then the spin label is attached to this specific residue. So one candidate is, for example, a cysteine or a tag, which is rarely used, in fact. Or you can think about, just as it was shown above, introducing a non-canonical amino acid, but of a simpler kind, the one that does not bear an unpaired electron, but very specific group, say unique functional group for the whole protein. And then you couple a spin label to this unique functional group. So regarding this first example, here are the 2 most used spin labels. This is MTSL, and this creates a double sulfur bond to the cysteine, like this, and then we have it. Or the malimidoproxyl, and this then creates the SC bond and saturates this carbon here. So this is then the spin-labeled fragment. As regarding here to the second example, we can think about phenylalanine modified with the azide group, and the kappa click here via the triple bond, and we get to this prof amino acid, or spin-labeled amino acid, that we also use in our labs. Okay, so this was a very brief review of the method itself, how it works, what is required, these unpaired electrons, in which sense they exist in the molecule, and how to introduce them. And by this, we have a tool at hand that is capable to work with non-crystalline samples. And in fact, with DEAR, distances are measured for samples that can be as a powder or a gel or in solution. And this is brings me to the next point, in solution. It can be whatever you have pH, there are other small molecules present. So it is also possible to apply the method to study things in the crowded media. Also, the size does not matter. This can be small molecules, large molecules, molecular complexes, whatsoever. So disregarding the size. And also inherently, because the method can determine multiple distances, it is possible to work with a mixture of structures. And having said all that, it is the method that is suitable to study intrinsically disordered proteins, because their structure is just the function of the environment, and they have to be studied in the physiologically relevant environment. Very well. So this was, so to say, the first part. In the second part, I would like to show you some selected application examples from our own research, and also to highlight the application possibilities of PIA. And the first example is here on the slide, which is called length. And apparently, if you have a method that allows you to measure the distance between 2 points, you can measure a length of something. And this particular example is from the research on gram-negative bacteria. And the question was here the acceptance of nutrients and transfer them from outer membrane to the inner membrane, and the energy transfer backwards from the inner membrane to outer membrane. So the space between the membranes is called periplasm, which is 15 to 20 nanometers. And in order to develop models for these energy transfer and nutrition transfer, the question was the following. There is this tone B, which is known to be essential, protein, which is known to be essential for this transfer. But the question was very clearly defined. Is the length of tone B enough to span the periplasm? So basically to take nutrients from here and bring it back there. Okay, because it is already research that is done, we can simply look up in the lab journal how we did this. So the tone B was expressed in E. coli, and there was a number of cysteine point mutants that were expressed. And after isolation and purification, they were labeled with MTSL. And just to show you again the scheme and the way the spin label was attached. So here are the experiments. This is how real oscillations of the spin echo look like with real experimental noise. And then from each of the traces, we can extract the distance. And now please look at these question marks. So here now the result appears. We measured a number of distances that basically cover the full length of tone B. And if we add up all the numbers, then we arrive at slightly above 20 nanometers. And by this, we deliver the answer. The length of tone B is enough to span the periplasm, and this should be taken as a basis to construct structural models. There can be also taken, we are now moving to the other example, a specific distance, a one single distance that is characteristic for the whole structure. And here I want to show you another example from the nucleic acid research now. And central here are the chromosomes and telomeres of the chromosomes that are important for or relevant for cancer and cell aging. So here at the telomeric ends, there is a single-stranded region of repeating units TTA, GGG, TTA, GGG. Now here is once again the short sequence that can fold to G quadruplexes. So these are very special structures, or they are called non-canonical structures, but in fact they exist, whatever you call them, where the guanines are arranged in the planes as shown here and schematically also here. And interesting about this is the very high and versatile topology. So basically this short sequence, depending on conditions, can fold in at least these four structures. And more so, if you have here a couple of nucleotides, more or less, the variety of structures grows. So what is actually the question here? What is the relevant structure out of those that probably can be targeted by medicines or whatsoever? Now, first we have to look, is there any specific distance that is characteristic for the structure? And you might think that there is one. And indeed, if you consider putting spin labels in these two positions, then you would expect to have different distances for all four structures, and they're also different enough to be distinguished with our technique. So let's look in the lab journal before we're doing this. Such a short peptide can be synthesized meaningfully by solid phase synthesis, and the spin label is used. This one is a deoxyuridine, bearing a nitroxide here. And then we performed this experiment, and we found out that in solution, there is not a single structure, but a coexistence of two, this one and this one. Fine with that. But if you remember, I said the couple of nucleotides on the left and on the right already influence the structure. But there can, in fact, in the reality, there are not the couple of nucleotides, but there is probably the second quadruplex. What is the effect of those? This is more interesting question, more intriguing. And you can think that the real experiment should be like this. Construct a long sequence that is able to fold three quadruplexes in a row and look at the structures of selected units that either has one quadruplex next to this or two neighboring. Yeah. And once we did this, we found that the one and only relevant structure is just the other one. And this is this one. And this is also for me a very nice example how you can selectively look at one unit within very similar or repeating units. So this was an approach to look at one single structure by one single distance. But having several distances, you can also map the global structure. And this example is now from the research on alpha-synuclein. This is known also as a Parkinson protein. And while its role is still unknown, it is related to Parkinson's disease, neurodegenerative diseases, and its interaction with membranes is crucial. So this is what is known so far. And it is an intrinsically disordered protein. So if you say to people like me, I have an intrinsically disordered protein, then I would say, let me label this, or let me give it to my students to label this. So let's look in the lab journal how we are going to study this. And this we do by creating a cysteine point mutants, expressing them in E. coli, and after isolation and purification, spin labeling with MTSL. And this work has been done. Now, this was the enema structure on my cells, but if you want to know the exact relative arrangement of these two helices, and the overall structure is called a broken helix for obvious reasons, yeah, then you can do like this. This is quite a number of experiments, but then you can identify the relative arrangement of the helices. This was on my cells, and you can refine it on small unilamellar vesicles, but this is in fact all about the curvature of the membranes. So this is the my cell, very small. Liposomes are somewhat larger, but if it is a bilayer, and the curvature is very small, like this, then the question arises whether this broken helix is the result of very high curvature, or it is intrinsic to the peptide itself. And this was studied. So here I show you the alpha-synuclein once again, the model for the extended helix, not broken here like with a loop, but it continues spanning 90 amino acids. And we were looking for these two positions, because here we can differentiate whether it is a broken helix, like here, or the extended one. And then, well, the protein is spin labeled. It is bound to the membrane, and we can measure this. And here is the result. We see here the distance distribution, and clearly two peaks that can unambiguously attributed to two conformations. And what we find here that indeed the curvature is the factor determining the structure. If you have a lipid bilayer with a smaller curvature than these ones, then the prevailing conformation is indeed the extended helix. It is 1:4 ratio here. Okay, so this was an example of how to map the global structure and to identify it from the mixture. But it is also possible to accurately locate a fragment within the large structure. And here is the example. It is the example from the research of mitogen activated protein kinases. So these are present in humans, and they are responsive for cell signaling. And their dysfunction causes then cancer or inflammatory diseases. What is known about these kinases, there are several types of them, and also structures are available, but they are available not for the entire kinase. They are available for the largest part, but not for this blue one, for this activation loop. It is highly disordered, and in fact, it is hardly seen or not really seen in the structures. Okay, so if something is disordered, let's put a spin label in there. This is fine. And where to put the second spin label? This is the question. And the approach is this one. Let's use the approach which is similar how the GPS works. We put several spin labels at different positions in the protein and measure distances to the spin labeled loop. And by this, we can locate this loop within the protein. So what is known about these loops so far? It is known that the conformation of the loop depends on the small molecule that is bound. It is known that some small molecules stimulate initiate the loop conformation where the kinase is active, and the others that it is inactive. There are also the names for the loop, which is DFG out and DFG in, but there is still no structure. And the structure is a key to function, this we all know. So the structure is still interesting, or at least the location of this loop. So what do we do here? Let's look in the lab journal. And now you already recognize that the protocol is the same. And by this, you can conclude that this protocol is the most widely used and the most well-established. And it is also very reliable. So expression in E. coli of cysteine point mutants, subsequent spin labeling with MTSL, and we are in business. Here are then some of the distance distributions that we measure always. If you look here, gray is for the kinase without small molecule, where the loop is highly distributed. And this distance distribution is very broad always. And if there is a small molecule added, it is a color, red or blue, then the distance distribution becomes narrower. And if we now try to combine all this distance distribution in order to locate the volume, we arrive at this result. So gray, again, the color coding is the same, is the volume where we find a distributed loop where no small molecule is bound. And once the small molecules are bound, then we have a smaller volumes in blue and in red, as shown here. And you see they are different. So we see immediately this effect of binding small molecules. The space of the conformational space of the loop is drastically reduced to a smaller volume. And interestingly, we find that for the active conformation, this volume is within the gray volume. And for the inactive conformation, this volume is located outside the volume, outside the gray area. So this is how even the distributed parts of the molecule of interest can be localized. and this also in solution. Now, this slide is called huge molecular complex, and indeed it is an example to study very large objects of interest. So this example is from RAC research, and RAC stands for ribosome-associated complex. This is responsible for functional folding of nascent peptides. And this is quite a large complex. It cannot be that easily studied. And what you do normally with large complexes, you break it down in smaller pieces and study them then. But here the research question was, what are the structural properties of this RAC complex and then associated with the ribosome? So full length RAC and then the ribosome on top. And this is not a trivial task, because RAC itself is large, but ribosome is just huge. If RAC is 107 kilodalton, ribosome is 3.3 megadalton. So this is a factor of 30, and factor of 30 is really something serious. If we take examples from our everyday life, factor of 30 we have in the weight between a newborn baby and an adult. Or it is the difference is the same as a motorcycle and a truck. And there are not many methods that can be applied for both. But I'm here to show you that deer can be applied for both, in fact. Let's look in the lab journal. This is, in general, the same expression of cysteine point mutants of E. coli and labeling. So here the selection was, or the choice was for this proxial spin label. And we are interested in this 4 helix bundle, because here where it goes, so this is the part from the RAC, and this is then the part that goes into the ribosome in the decoding center. So then the distance measurements were performed, as you see here, and then by combining them with molecular dynamics, we were able to constrain the structure, this 4 helix bundle as shown here, to derive a model and also to prove that this 4 helix bundle is extremely rigid, even if it is, if RAC is bound to that huge ribosome. Now this does not change its structure significantly. At least with our technique, we can just prove that the structure is rigid, not that there are any changes. Okay, so huge molecular complexes can be studied. So we come slowly to the point where molecules can be studied in complex environments. For example, membrane proteins in membranes. So here is the example I've prepared. And this is the example of ethylene receptor in plants, which is responsible for ripening of plants, accession, and so on. And in spite of this, of the importance of this receptor, there is still no structure available. The only thing what is available is this ab initio prediction. And this prediction was existing there for a couple of years until very recently another prediction came out from the AlphaFold. So AlphaFold is the program from Google and based on artificial intelligence. This is the second structure, and you see the first helix is coinciding with the ab initio prediction, but the other two are not. So now we have 2 structures, and we have a question. What prediction is correct, and what is relevant? Now, and this we are going to answer with our technique, with the deer spectroscopy. And the approach that we use here is again expression in E. coli of cysteine point mutants, post-synthetic spin labeling, and then reconstitution of the spin-labeled proteins in large unilamellar vesicles. So this is our model for proteins. Here again, one of the models, and these colors show all the positions that were labeled. And in first instance, we are going to look at the distances within the helices to check whether the helices exist. So here is the experiment. Let me explain the colors here. So the red, blue is the distance distribution as measured in our experiment. And in red, full line or dotted line are the predictions from ab initio model and AlphaFold. And if you look at all the three distances, the widths are different, but still they lie essentially within the same range. Yeah, so by this, we say, fine, the helices also exist and both models predicted them correctly. But then in the second part, we are going to look at the distances between the helices. And here is the result. And we are not going to analyze in detail all the distance distribution, but just catch one. And it is the red dotted line, which is the AlphaFold prediction. And as you see, in most of the cases, it just stands apart. It does not coincide with the experiment and with the other model. So this was the experimental finding. And by this, we deliver the answer to the question that rather the ab initio prediction describes the experiment than AlphaFold. But it is nothing bad about this. I think it is also an important contribution in order to improve these artificial intelligence based models for prediction of protein folding. Okay, and now the environment in which we measure becomes more complex and complex. It was a huge molecular complex. These were membrane proteins, and in fact, the environment can be as complex as a cell. And here is an artist's impression of a cell with many things inside, and this is what is visible in the EPR spectrometer. And in fact, it is nothing because the cell is entirely diamagnetic. But here is our vision. If we have a spin labels, we do see them, and we can also measure distance constraints between them to locate them with respect to each other. And if we combine now this with some preliminary knowledge about the protein of interest, we can see it among all other compartments and players within the cell. The experiment I want to show to illustrate this is rather a proof of principle experiment in which we were interested in the conformation of a polyproline chain. Yeah, so in the lab journal, we have the following: a solid phase synthesis for a relatively short peptide bearing a lot of prolines and 2 cysteines at the desired positions. Then we couple a ligand to the cysteines, in fact 2 ligands, and we load it with gadolinium. So here the spin labels, and also to have some variety in my talk, is not a nitroxide anymore, but a gadolinium-based one. Now we have this, and we micro-inject the solution of this peptide into cells. The cells are Xenopus laevis oocytes, and the micro-injection volume is something 30 nanoliters or so. And then we have it inside the cell. We let it incubate there for a certain time and distribute. And here is the example, the result. We have the oscillation of the spin echo. Here is the experiment. And in green, we have a distribution with 2 peaks. And from this, we conclude that there is a coexistence of 2 conformations of this polyproline peptide inside the cell. Yeah, so these were the examples from constants. And now let's make a short break, link back and see what is constants and get some impressions from there. So we are located in Germany, other side of the ocean. And Constance is a small city at the very south of Germany, next to the Lake of Constance and direct on the Swiss border. If I'm going to zoom in here or just show you another map, then this is already the whole city with 80,000 inhabitants. So this is the whole city. This is already the border to Switzerland, and everything to the north. These are the buildings of the city of Constance. And the university is not there. They decided to put it a bit aside. So it is here in the forest, isolated from the city, and the city is isolated from the university. This is now the university, shooted from the air, and we are located with our instruments, with our experts, with our people, just here in the middle in this propeller-like building. And so here is some impressions from our lab. We have very high-tech electronics to perform this, and of course, many computer, or you need at least one indeed to operate the spectrometer. This is the micro-injecting device here, and the cells are somewhere there. We work at cryogenic temperatures, and the things get frozen sometimes. We always have help from our dear colleagues assisting, building the system together, and we have a lot of samples to be measured. Okay, now back to the topic. Here I decided to the very end this would be an important thing. And the slide is called width of the distance distribution. Probably you noticed that with all the results I showed you, there was not a single one with a simple distance. Rather, these were always distance distributions. And one has to take it into account that they exist and to understand them properly in order to make conclusions about the structure. So I would like now to talk about these widths. There are just 4 cases where they can arise from. The case A is the spin label is covalently bound to the protein via flexible linker. So because of the flexibility of the linker, the spin label can take on different spatial positions. And if we are about to measure the distance from this letter A to the nitroxide, then it will be different depending on the spatial position. And this causes, of course, the measured distance to be broad. Another case is, suppose the link is rigid, but then the backbone can be flexible. This results exactly in the same thing. The spatial position of nitroxide can vary. And if we measure the distance from now letter B to the nitroxide, then we measure slightly different distances. We look at many molecules, and each molecule has then different distance. And then we have again the distribution. If we occasionally look at the object that exists in the equilibrium where the spin label parts happen to have different distances, we again have a broadening. And finally, if the molecule itself is extremely flexible, then we measure distance between different spatial points, and this causes also the distance to be broadened. Many cases you can take into account from other experiments or by designing the positions where the spin label can be placed. Regarding this one, this can be calculated. And the way we do this, I would like to show and explain. So this now again highlighted the backbone of the peptide, and cysteine is labeled by MTSL. The covalent linker is here. It has 5 single bonds, and the rotations about each of the bonds are possible. So what we do now, we start molecular dynamics and look at all these possible rotations, and we perform a statistics for the angles that are preferred. Well, the rotations are possible, but there are angles that are preferred. So 4 bonds and then 4 angles, 1, 2, 3, 4, and 5. So this circle is to be understood as 360 degrees, and these flowers are the preferred angles. So you see apparently there are not angles are equally energetically preferred, but only some of them. And then we can combine and build a library of rotamers or conformers, the library of rotamers we call them, and we know all the spatial positions of the spin label. And now let's go back to the example of the kinases, you know, where we located the distributed loop. So the real situation is this one. Here is the spin-labeled loop, and we localize it not against the point, but rather against the cloud. So the distance that we measure, or the distance distribution, are the collection of distances from the label to all the red dots here, weighted by corresponding weights as they are represented here in this cloud, and all the same here. And now with this in mind, let's recall again our in-cell experiment, which I described only briefly. But now let's analyze and understand the result. Or in other words, let's interpret the result. So this is the oscillation of the spin echo. In green, we have the distance distribution, the experimental one. And now we are going to perform calculation of the rotamers. And we take two known structures for polyproline helices. This is type 1, a compact one, and type 2, extended one. And then we calculate rotamers. Red dots are gadolinium ions. And apparently in the experiment, we measure not a single distance, but the distances between all the red dots weighted by their corresponding weights. And then if we take one-to-one mixture of these two distances and plot them in orange here, we get a pretty nice description of the shape of the distribution. And of course, the peaks are nicely reproduced. So this is exactly the point. We do measure a broad distance distribution, but this is still a hallmark for one single well-defined rigid structure, as shown here and there. And with this, I'm already at the end of my talk, and I can only sum up that it was about DEAR, that this DEAR technique was presented as a tool for structural determination. It has its advantages and disadvantages, any technique, but let me highlight advantages, of course, again. It is capable to deal with non-crystalline structures, with the samples in solution and also in the crowded media. The molecules disregarding the size are all suitable for the technique, and technique itself can cope with a mixture of structures and identify them. Not only identify, but also quantify the relative weights. And this is also the technique of choice when it is about studying intrinsically disordered proteins. So these were the points that I highlighted in my talk. But once you start working with this, there is even more about this. We can determine labeling degree, and depending on the labeling degree, you can study how different sites are accessible to be labeled at all. We can measure local concentration of spin-labeled things. We can measure spatial distribution, be it 3D or 2D, depending whether the molecules are located on a membrane, it will be 2D, or equally distributed in solution, which will be 3D. We can monitor aggregation and binding events simply by looking at the distances as they appear and disappear. So this was the work entirely done in our group. I mean, no, the application example, of course, not the development. And here is the recent picture of our group and all the collaborators here. So the credits also, of course, goes to them. And with this, I'm now really at the end. And I thank you very much for the invitation and for your attention.
Thank you very much for a very nice talk. I definitely learned a lot. I have a very technical question from the beginning, and it has to do with the equation that you showed, where the frequency is proportional to 1 over r cubed.
Yes.
And so this ties up with what you showed us at the end, that you have the spin labels going around like crazy.
Yep.
So shouldn't it be 1 over r to the 6th, given that you have a dipolar interaction and that goes as 1 over r cubed? that I agree. But if
Okay.
these guys are rotating freely, with one with respect to the other, shouldn't you have a Boltzmann average of that, and that gives you 1 over r to the 6th?
It is a bit differently. So I stressed many times that we study something in solution. So we do study something in solution, but it is then frozen. So what we look at, it is a frozen solution. It is then still a powder. So there is no free rotation during the experiment. There is also no averaging of the dipolar coupling during the experiment. So what we look at is the billions of molecules frozen in whatever instance they were caught by shock freezing. And by this we look at the conformational ensemble that represents what we expect at room temperature and in solution with tumbling around and so on.
Okay, I agree that you flash freeze them.
Mm-hmm.
But then again, given that you have an ensemble measurement, you're going to have some interactions where the orientation is parallel, somewhere it's like this, somewhere it's like this.
Yeah.
That doesn't matter?
It does matter. Now, let me see if I have this. The equation that I showed is simplified. There is an angle dependence, and now, exactly, so it is here.
Uh-huh.
Probably this is your question. So
yes.
We have the pair of spins, and there is an angle relative to the direction of the magnetic field. So, and there is additional this angle factor in there, and yeah. So this I kept for expert because then you don't have one single frequency, but rather a pattern of frequencies that we then analyze.
Okay. But in the end, what matters the most is, what you care for the most is the distance dependence, not the angular dependence.
We don't care about the angular dependence because for the amount of molecules we have, we can assume a random and equally distributed this angle. That's why a simple equation.
Okay.
But if there were something else, the processing becomes much more complicated. It's still possible, but complicated.
I see. Along the little bit same lines, when you were telling us about the quadruplexes in DNA.
Yes.
Where you had this very nice slide with the 4 possible structures and told us that there were many others. In the end, you chose one of them as the significant one.
Yeah.
Because you had this long stretch where you had 3 of them in tandem. So does this happen because they stack one upon each other? And that is why you get one of them to be representative in solution?
So this I don't know. In fact, it was even my own research, but as the spectroscopist, I was only interested in what is the structure. Why is the structure like this? Actually, I don't know. And I think it is not that easy to conclude on, because let me think. Yeah, probably. Well, from this experiment, we cannot identify the reason. But now, because of your question, one might think of designing another experiment to label 2 different quadruplexes and then see whether they stack or not. Because then by combining this with molecular modeling, one can already expect some distances for stacked and non-stacked and see what happens. Yeah, so there are more experiments needed in order to answer your question.
Right. You know, I see that. Well, I do molecular modeling for a living. So whenever I listen to talks like yours, I'm already thinking of what I would need to do in order to finally to get to the real deep reasons why things happen the way they do.
Yeah, but I think the combination of the experiment with molecular modeling will deliver the answer as to relative orientation of the quadruplexes and probably their interaction or impact on each other.
Right. So thank you very much.
I have a question in this slide. Now, based on the distance distribution that you show in the other slides, I cannot understand how it's possible to discriminate between 2.9 and 2.4. I mean, again, with the simulation and just putting the motion of the labeling, I think that your distribution is going to overlap in those two, or not?
So here I did not show any experimental distribution, but these are published works, and well, they do overlap because the distributions are broad, but you can still identify peaks. Okay. And this is already enough. So if they were coinciding, this would be really bad luck. But they can be still discriminated.
I have another question, but I think you already answered me. But when you were showing the different distance distributions and that you were measuring different populations, just pop up if there was a way in which you can transform that to some kind of dynamics. But I guess just because the temperature in which you are working, that's kind of difficult.
So the sample is prepared at room temperature and shock frozen in liquid nitrogen. And we assume that we trap all the conformations that are there at room temperature. There is no dynamics during the experiment, but we look at the snapshot, in fact.
Yeah. Okay. Thank you. Very nice.
Thank you.
Mikhailo, sorry, is the chemical environment a factor to measure your measurements? I mean, how the buffer that you choose or the salt that you add affect the measurements?
There is no effect because we look entirely at the unpaired electrons. So what there are technically or experimentally in the lab, there are only two problems. Now three. Okay. So three. The first one is that we now let's say this would be the third one. The first one, if the spin label gets reduced in the medium. This is really bad because then we have nothing to look at. The second problem is if the linker gets reduced in the medium and the spin label is detached. Then looking at this has, again, no meaning for us. So these are the two problems that we try to address while preparing the experiment and designing and looking for appropriate spin label, appropriate linker, and so on. And the third problem is of a very technical kind, but it is still relevant. We need to add vitrification agents, some cryoprotectants like glycerol or ethylene glycol, so that once we freeze the aqueous solution, it forms a glass. It does not form crystals, because in crystals we have an inhomogeneous distribution of spin labels, and it obscures, in fact, the measurement. So we always add a bit of glycerol, which is 20% by volume as a rule. And this is the way these samples are prepared.
Okay, but for example, I'm a protein crystallographer, and in our case, the reduction source are the X-rays by itself. But in your measurements, which factor may increase the propensity of the label to be reduced?
Oh, in this sense, so the measurement itself is harmless, absolutely. It is only the chemical environment. As long as the sample exists in solution, if we have whatever reducing enzymes, ascorbate, for example, it it destroys the spin label. But the measurement itself, it is no problem at all. So for the measurement, we require low temperatures, microwave irradiation, and magnetic fields, and none of those does anything to the sample.
Then, in principle, if you are working with a very pure modified protein, you are not going to have any problem. The problem is when you are working with a complex sample as a full cell or something like that, isn't it?
Yeah. The problem only if the spin label gets reduced or damaged. And another problem, so if we're talking about application, is that inside the cells, the amount of spin labeled things is very low. So the concentration is low. And our problem is then also the sensitivity, because in these in-cell experiments, we already measure at the limit. In vitro samples are very nice to work with, to prepare. You can concentrate them up and prepare test or provide textbook data of utmost quality, but in cells are not. And one of the factors is very low concentration.
Okay, thank you. And finally, which is, for an average protein, which is the concentration that you need to have a good measurement?
So we need a 20 micromolar solution, and we need 50 microliter of it. So this is the sample that is required. We can do with, say, 2 micromolar as well, but this would be challenging.
Okay, thank you very much. I really enjoyed your talk.
Thank you.
Nina?
No, what I was going to ask is if there are any questions in YouTube from the people that were watching the seminar, or from Facebook Live.
None in YouTube.
Sandra, do you know if we have any questions from Facebook? You can always ask them in Spanish and we translate. It's not a problem.
The problem is going to be our translation.
Yes, there's always hand waving. It's very useful. When you cannot find the exact word, it goes like this. It's fantastic. Okay, it seems that the rest of the audience found it very clear.
Which is also fine.
Yeah. Lina, no? No.
Yeah, I mean, I have a lot of questions, but I don't think that we have time for that. I wanted to ask, for example, the aggregation. For aggregation, I have read that you can use just CW-EPR. I mean, you use your spin label and then you follow the aggregation by CW-EPR. But I don't know, this is not too related to your talk, but maybe you can tell us something.
Well, I can tell something, of course, but it is indeed not related to the talk, and I don't have a single figure to show you. And this I find a pity, because just showing with the fingers and pantomimic how these spin labels move is probably a possibility to do, but not the way to do this. So within EPR, there are other techniques that can be used, and the one is called CW, which stands for continuous wave. There we look at the motion of spin labels in solution. So it is now really in solution without any tricks, without glycerol or whatsoever. And the spectrum that we record is sensitive to the motion of the spin label. And the motion is, of course, governed by the, say, in simple words, by the size. So if the spin label, the small thing, is tumbling in solution, it goes very fast like a fly. And if it is attached to the larger molecule, say an elephant, it tumbles slower. And this we can see in the experiment. And then probably, Lina, what you address is if we monitor aggregation, then we basically see how the object that bears unpaired electron becomes larger and larger and larger. So this is what happens in the sample. And what we see then, that the rotation, this tumbling, becomes smaller and smaller, or slower and slower and slower. And this we can see in the experiment. Yeah. And then from these data, we can determine this rotational correlation time as a constant to characterize what we see. And we can also calculate the hydrodynamic radius of the object that tumbles, be it fast or slow.
Mm-hmm. Nice. Thank you, Misha. Thank you so much. Very nice talk.
So this last experiment that you described gives similar data to fluorescence and isotropy measurements. No? It would be complementary to fluorescence and isotropy, because there you also get the rotational tumbling times. But in that case, of the fluorophore.
Yeah. I had to concentrate myself a bit because this is not the method I work with, but just I've heard. Yes, it is similar in this sense. So because you get this correlation constant, right?
Mm-hmm.
Exactly, yeah. Right. So this is related.
So for me, it's nice to have different ways of asking the same thing, and then to make them come to a reasonable conclusion, then it means that maybe you've got something right. No?
Yeah, exactly.
Okay, so I don't know, Lina, you drive.
Well, I don't know. What do you usually do?
Just thank our speaker.
Thank you. Thank you so much, Misha. Thank you.
Yeah,
Really.
You're mostly welcome. Well, I also have to thank for the invitation and that you allowed me to postpone it by 1 week because I could not make it last week. And for the very warm taking at the very beginning, for the very nice small talk, as though we know each other already for months or so. So it was my pleasure to present for you, to say something, and also my pleasure to answer questions from really people that are interested in the subject. So also thank you from my side.
Okay, so I think that this is it, right? So we just say goodbye. Yeah. So I hope to see you soon, Misha. Thank you so much.
Hope to see you here.
For invitation.
Thank you for your time and for your talk.
Thank you.
You're welcome. All the best. Bye-bye.
Bye.